So I think I will just start the presentation, then people will join usually. So if anyone has any doubt or anything in between, you can unmute yourself and ask the question. Okay. So I probably want this section to be informative as soon as possible. So I think the slides are visible, right? Hello.
Yes, sir.
Okay.
Yes, sir.
Thank you. So good evening, everyone. My name is Isaac Cherian, and I will be taking my NPTEL course on topic TAship on the topic cell culture technology during this time on Sunday from in the evening section of about 5 p.m. to 7 p.m. Today I will be taking the sections on week 0 as well as week 1. So I think week 0 should also be taken, even though it's a practice section. It might be important to you also. So, but I will start with the week 1 section, and with time I will continue on to the week 0 section. So coming on to the first question. So for the first question, it states that in which phase of the cell cycle the cell either progresses towards DNA synthesis and another division cycle or cell division cycle, or exit the cell cycle to commit to differentiation. So if anyone knows the answer to the question, would you please answer? Okay. G1. Anyone has any other answer? Okay. Okay. So the answer to that first question is probably the G1 phase. So I will just give an explanation to that particular answer. So this is the particular cell division type. You can see the diagram, right? So this is the particular the cell division cycle that takes place in a particular organism. So there are 4 different types, right? So M phase is there, G1 phase is there, S phase is there, and G2 phase is there. And M phase is probably the phase where cell division takes place, right? And it consists of about 4 cycles, most probably 5. And the stages consist of prophase, metaphase, anaphase, telophase, followed by cytokinesis or cytoplasmic division, where the cell divides into 2 progenitor cells, right? So once this is complete, cell division is complete, the cell can enter into a stage called as G1 phase. Okay. So once in the G1 phase, the cell undergoes cell growth. So once cell divide, it needs to undergo cell growth. It needs to size up its volume so that it can prepare for another cell cycle division, right? So once in the G phase, it can enter into, there are 2 phases differentially. So one is the S phase, where the DNA replication take place. It is also called as synthesis phase. So in order to enter into a S phase, in the G1 phase, the proteins required for DNA replication need to be formed within the G1 phase itself, like proteins required for replication and all. And or else, other than the S phase, the cell can enter into another phase called as a G0 phase or a quiescent phase. So most probably the cells at this stage gets differentiated to perform specialized functions. Suppose that in case of a mitotic phase, the cell has given about a stem cell has given about 2 progenitor cells. So one progenitor cell enter into S phase while the other enter into a G0 phase. Suppose that it produces a particular hormone. It's a hormone secreting cell, right? So that is specialized in that particular case. So that particular cell commits into a differentiation stage. So in other stages, in the subsequent stage, it can also it can also enter into a G1 phase and into a S phase, which I will discuss in the later presentation also. That is the answer, basically S phase. So I hope this answer is clear, right? So anyone has any doubt regarding this? Hello, does anyone have any doubt regarding this?
No, sir.
Okay, so I think it's clear. So let's move on to the next question. So this is a basic question. Basically, most of you might know about this question. So which of these is not a constituent of animal cell? Cell membrane, cell wall, nucleus, or DNA?
Cell wall.
Cell wall.
Cell wall.
Cell wall. Okay. So that's a basic answer. So basically, if you look into animal structure, you can see that these are the different components. There are different organelles present inside there, right? So basically, animal cell lacks this cell wall. And could you please give an example of another type of kingdom or any other species that where cell wall is present?
Fungi, sir. Fungi.
Fungi and one more.
Uh, Monera, bacteria. Bacteria.
Okay, cell wall is present there, and one more. And important, like it's close to animals. Plants also, right? Plants also composed of cell wall, because the cell wall is a basic protective agent. It is found that it is important to regulate the structural integrity of the cell. So even though plants are immobile, so they are most probably affected by climatic changes and other infectious agents, right? So in order to provide additional protection, this cell wall is essential in case of plants as well as in fungi as well as monera, right? So that's also an important question, and also to maintain the turgor pressure within the cell wall, to maintain the structure and rigidity of a cell within the plants, the cell wall is important. So I hope this question is clear. Anyone has any doubt? So does anyone has any doubt, or I will proceed on to the next question then.
Yes, sir.
So this is the next question. In which phase of the cell cycle DNA replicates? So could you please provide the answer to the question?
S phase.
In S phase, the DNA replication happens, right? So I will just move on to it. So once the cell has undergone mitosis, the cell has divided into two progenitor cell. If you consider an individual cell, then it undergoes pass through a G1 phase where it can move into G0 phase, where it comes to differentiation, or else it can move into the S phase. So before S phase, they produce the proteins that are necessary for DNA replication to happen. So in this phase, S phase or the synthesis phase, DNA replication happens. And afterwards it moves into a G2 phase where it prepares for the mitosis. Once the checkpoints are passed, it moves into a mitotic phase, which is where the prophase is the first phase of this cell cycle division, right? Okay. So anyone has any doubt regarding this?
No, sir.
Okay. So I think it's clear. So next, coming on to the next question. In RGD, G stands for? So could any one of you answer?
Glycine.
Glycine. So this is basically an amino acid, right? And R stands for? Could you please give the abbreviation for both R and D? Basically, glycine is amino acid. This is a tripeptide, basically. So, coming on to two other amino acid, could you please give an answer? R stands for?
Arginine.
Arginine, and D stands for? D is one of the amino acids, right? So I will just give you the answer. So answer is glycine for G. And for I will just give you a diagram. Basically, RGD stands for arginyl glycine aspartic acid. So the R stands for arginine, G stands for glycine, and D stands for aspartic acid. So you should know the basic abbreviations of each amino acid, because it will be essential for you in the future also. So because there are different amino acids, right, the abbreviation might change in between, so you might not know the proper answer for that. So basically it's a tripeptide, and it is responsible for cell addition to extracellular matrix. Basically you can see that extracellular matrix is what keeps the cell intact with each other, right? So basically and it is also responsible for the cell to grow in a particular that culture plate, right? Though if we provide an extracellular matrix, then we culture the cell above it. So that particular extracellular matrix plays a role in maintaining the addition of the cell. So and apart from this, these are part of other proteins like fibronectin, fibrinogen, fibronectin, etc. These are also extracellular matrix proteins, and these proteins have this RGD motif which binds to these proteins called as integrin. These are basic receptors basically, and upon binding, this integrin receptors gets activated, which in turn produces signal transduction there, which helps in cell proliferation, cell migration, etc. because it's an extracellular matrix protein. So this is an essential component of this signal transduction cascade. So I am not going into detail in this. I just show a diagram for your knowledge and all. So in this case, RGD, G stands for glycine. So anyone has any doubt regarding this? Okay, so I will move on to the next question then. So dash implies what implies that the specialized property of a cell are lost irreversibly. So anyone knows the answer to that question? You can just type it out, or you can unmute yourself and answer that question. So one question is D, option D, differentiation. Anyone has any other question? So okay, D.
The differentiation.
D differentiation, or could you please repeat? Differentiation or D differentiation?
Yes.
Differentiation.
D differentiation. So the answer is D differentiation basically. So basically I am showing you just a diagram in this case. So what is when a cell suppose that there is a stem cell. So it has undergone several cell division cycles. So if a single stem cell is there, it gives rise to two different progeny cells, right? So suppose that either both of them, or either one of them is getting is converting into a cell which is specialized to carry out a specific function, right? So this process is called as differentiation. So what happens if the differentiated cell in turn becomes a stem cell which loses its specialization? So this particular phenomena is called as de-differentiation. And basically if you see de-differentiation and all, once it is de-differentiated, it cannot revert back into a differentiated stage. The previous differentiated stage it cannot possess, but it can later transform or redifferentiate or transdifferentiate itself into another cell which carries out a different kind of specialized function. So this term you have to see irreversibly, because in case of other term is there de-adaptation. In de-adaptation it is kind of reversible in nature, because it can this particular de-differentiated cell can revert back into a differentiated cell which has the same functionality that it had before. So the answer to this question is de-differentiation and not de-adaptation. So you have to look into the difference in this case. So I think, yeah, and either the de-differentiated cell can enter into a cell death stage, it can die off, or it can enter into another cell cycle phase, and if provided that specific cues are provided, right? So that's a different kind of phenomenon in this case. So I think that particular answer is that particular doubt is clear, right? So anyone has any doubt or any other question, or is it clear?
Clear, sir.
Okay, so that's the difference between the one is reversible, other one is irreversible in nature. So even though the terms are kind of same, but there is some differences in between that you have to look into. So cardiac cells are mostly next question is the question. The cardiac cells are mostly categorized as, first option is bone cell, neuronal cell, muscle cell, and epithelial cell.
Muscle cells.
Muscle cells.
Or another term scientifically, what are these cardiac muscle cells called as? Scientifically, if you give the answer.
Cardiomyocytes.
Cardiomyocytes, right? So cardio means cardiac, myo means muscles, and cyte means cell. So that's a probable answer to this question. So basically these are connective tissues, right? They are connected together by an extracellular matrix and all. So the answer to this question is muscle cells or cardiomyocyte. So coming on to the next question. So this is a common type of question, you might know this particular cell type, right? HeLa cell line was derived from which type of cells? First one is muscle cells, second one is cervical cells, prostate and neuron cells.
Cervical cancer cells.
Cervical cancer cell. Okay. So I think everyone has the same question here. Answer here. So answer is cancer cell. So basically this was a first cell line, that cellular model that was ever developed, and it was developed by John Hopkins Hospital, right? So the cells were derived from cervical cancer sample from a patient known as Henrietta Lacks. You should know this answer because the HELA, the abbreviations, that is HeLa, is derived from this particular patient, that is Henrietta Lacks, in 1957. And it was first donated by these two scientists, that is Theodore Puck as well as Philip Marcus. So these are the first human cells to be cloned also in this case. And later, in case of cancer research, not only in cancer research but also in drug discovery and other causing conditions and all, stem cell research, and also these HeLa cell lines are at most used also. So but the problem is that with the HeLa cells is that the HeLa cells were actually taken from the patient without her consent. After the patient passed away, the HeLa cells were acquired from the patient, and later it was used for different purpose like research purpose and all. So without the consent of the patient, actually the cell lines were used. So there were several ethical issues that are related to that, because ethical consent should be there when we are using anything for our biological research purpose, right? So even if without the consent, the genomic information of the patient was published within the NCBI database, so that was quite an issue there. But with the utmost use of this particular cell line, several research, even several medical breakthroughs, as well as about different patents, nearly about 11,000 patents were filed with the help of this cell line. So afterwards, several other cell lines were also used, HEK293 cell lines, CHO hamster cell lines and all. You might know about this, and you can also get to know much about other cell line also. So I think the answer to this question is clear, right? Should I move on to, anyone has any doubt, or should I move on to next question? Does anyone have any doubt?
No, sir.
Okay, so I will proceed on to the next question then. So next question is which of these gases along with its buffer is used to maintain the pH in culture medium? First is oxygen.
Carbon dioxide.
Yes, hydrogen, argon. Carbon dioxide is the answer, because in the NPTEL course that my minus has taken, you have came to know that carbon dioxide actually interacts with the water that is present within the cell culture media, right, to form what is called as a carbonic acid. So this is the stage, right? So this carbon dioxide that is present in gaseous, we usually provide that as a, even the cell actually respire out carbon dioxide, and also the incubator that we are keeping the cell culture actually is provided with the carbon dioxide from outside. So this is provided as a gas there, and it mixes with the aqueous environment that is present within the cell culture to provide what is produced, what is called as a carbonic acid or H2CO3. Later in aqueous phase, it gets divided into these two ions, that is bicarbonate ion as well as protons. And this proton actually is the what is a pH basically. So pH is dictated by the number of protons that are present, protons or OH minus, that is hydroxide ions that are present within the media, right? So if protons are more, then pH will be more probably much less, as less than 7. That is, it moves on to acidic environment. If pH, if protons are less as compared to that of OH minus groups that are present within the media, then it will more probably a basic group. So based on the level of this proton that is present in the culture media, the pH can be fluctuated. Most probably at this range about 6 to 8 pH, that range can be maintained there. So that's the buffering range when you are using this as a buffer, that is gas as a buffer in the culture media. So this particular buffer, right, this is important when you are maintaining a cell culture media, so this carbon dioxide bicarbonate buffering system. And even in the physiology, in our individual physiology also, in our blood, this also play a major role in the buffering, maintaining the regulating the buffering capacity, and that is essential in case of our individual livelihood and all. So the combination of this carbon dioxide along with the bicarbonate ion is an important buffer in extracellular body fluids also. And the incubators provide this carbon dioxide rich in atmosphere, right? After we have did the culture and all, we are incubating it in the incubator which have this carbon dioxide rich atmosphere, and which provides this buffering. So the media that we are providing, right, the media where the cell culture is grown has this carbonic acid salt into it, because the carbon dioxide is provided with the incubator and it mixes with the total environment, giving this kind of buffering system so that the pH fluctuation can be maintained within a particular range. So that's why this particular gas is important, especially if you are using this kind of buffering system to maintain the cell viability or incubating the cell culture at most. Okay, so anyone has any doubt in this particular question? It's clear, right?
Yes, sir. Clear.
Yeah. Okay. So next question is, what may implies that the synthesis of specific products or other specialized functions are under regulatory control and can be reinduced if right conditions are provided? This question I also have some doubt with, basically. I would like to see what answers you provide, basically. So anyone can type, or you can just turn on your mic and just tell the answer. Okay, one answer is I got is the de-adaptation. Another answer. Anyone has any other answer? You can freely like comment the answer, basically de-adaptation. Okay. Any other answer? Differentiation. Okay, so there are variable answer to this question, I suppose. So basically it implies that synthesis of a specific product or specialized functions are under a regulatory control. So basically, and can be reinduced if right conditions are provided. So basically it means that from the term reinduced, so it can either be de-adaptation or de-differentiation. But in this case, differentiation I am not sure, because differentiation means that the cell is already at this phase, it cannot be reinduced or readapted back to a specific specialized function, right? It should have been in a differentiated phase. Later it should have moved into a de-adapted phase or a de-differentiated phase, so that it can be induced by certain regulatory control, so that the so that it can be reinduced to provide that specific specialized function. So I am having a doubt whether it is de-adaptation or de-differentiation, because it haven't said whether the specialized function is the similar to which it has exhibited earlier, or whether it is a different function. Whether it is a different function, it means that it is irreversible in nature, then it coins most probably a term de-differentiation. Where if it is reversible in nature, it can be de-adaptation. It is at a de-adapted stage. So if any regulatory control is there, then it can be brought back into a right condition, or it can be brought back into a reinduced into a specific specialized function that it has before. So so answer can be either be 2. So if anyone has any question, I am also doubtful in this particular answer. So anyone has any doubt in this case, or anyone has a proper explanation for this, please unmute yourself. You can freely speak. So I will just I have put the answer as de-differentiation here. So this is the whole diagram, basically. So anyone has any doubt? Okay, so I think we have to wait for the answer key probably for this also. So but if anyone has any doubt, please put a query in the NPTEL site also, so that we can get cleared about the proper option for this answer, or the proper option for this question, sorry. So I will just move on to the next slide then. So which of these is a non-Hadron cell type? Osteoblast, hepatocyte, erythrocyte, or neurons?
Erythrocytes.
Erythrocyte, right? And anyone has any other answer? I think this is a basic question, because in the first slide itself are actually talk about this answer. So okay, majority of the answer is erythrocyte. So the answer is erythrocyte, basically. So I will just show you an image of certain of all these options, basically. This first one is the osteoblast. So osteoblast in bone cell, basically. Bone cell actually have two types, osteoblast as well as osteoclast. Osteoblasts are the bone forming cell, where osteoclasts are the bone destroying cells. So there is a synchrony between creation as well as destruction in this case. So osteoblast form bone cell, osteoclast destroy bone cell. This for just for an additional information. So this is a microscopy image, a good microscopy image of the bone cell given here. And next one is the hepatocyte. So basically hepatocytes are the liver cells, right? So, and you might know that also. And erythrocytes are also called RBC. So, this RBC, you can see that these are separated from each other because it has to move from one place to another through blood vessel. So, if it is connected to each other, then it will not be performing well, right. So, it has to be separated from each other. So, you can see that the RBCs are separated from each other, they lack an extracellular matrix and also the RBCs lack a nucleus. So, it cannot divide any further. So, it is specialized in that function actually. So, it is it lacks the extracellular matrix. So, they are most probably a non-adherent cell type. So, this is the next one, that is neurons. You can see the neurons here actually connected to each other. You can see different kinds of cells, right. Neurons are adhered to each other because they should have that particular I think someone asked that question whether neurons are adhered to each other. So, neurons are basically adhered to each other. They should be connected to each other so that the electrochemical impulses should travel from one region to other, right. And they are also forming a part of the brain. Brain is actually tissue. So, neurons are forming majority of the tissue what forms a nervous tissue. So, they are also kind of connective tissue in this case, and they also have an extracellular matrix. Because of the presence of that extracellular matrix, they are called as a non-adherent cell type. Yes, they are connected by synapse, but individual neuron, the synapse is actually a part of that particular neuron, right. So, they are connected. The synapse is a junction that is particularly present between two different neurons, so that the electrochemical or the neurotransmitters flow from one neuron to other. So, it actually is a connective tissue, and base of a connective tissue, it has an extracellular matrix there. So, I think so it's a type of a non-adherent cell type. So, erythrocyte lacks that because they have to move from one place to another. So, that they should be in an individual cell like manner without adhering to each other. So, any other doubt? So, is that doubt clear, or any other doubt is there basically? If anyone has any other doubt, you can
Yes,
just
sir.
it's clear, right? Okay, so I think it's clear. So, I will just move on to the next question. So, I will just start with the next week question. So, week 0 question basically. So, do you have any extra doubts or anything that came up your mind in between regarding the previous week 1 questions in this case? Any other question that might have came up during the presentation and all? Okay. So, okay, so you already typed the question answer. Okay. So, PBS stands for here phosphate buffer bovine saline. Phosphate buffered saline, right? So, some of them types option C actually. So, PBS stands for phosphate buffered saline, right? Okay. So, basically it's a buffer system, right? PBS is a common buffer system. Even if you are carrying out any particular type of activity, any kind of any other techniques where you have to wash out the cell, or any other type of enzymatic activity, microscopy, any other type of FRET analysis and all, basically you wash the cell with what is called as a PBS, right? So, it is also called as phosphate buffered saline. So, from the name itself, you can actually come up to know about the components of some of the components of the buffer, right? So, can you please state some of the components of the PBS, that is phosphate buffered saline? Some of the components that come up to your mind when you are just looking at this name. Phosphate buffered saline.
Sir, phosphate and saline means NaCl.
NaCl is also called as salt, right? So, that's why it's called as saline. And the phosphate is provided from, I will just show you some of the information. So, it's a buffer solution that is used commonly in several biological research and all. So, PBS is actually isotonic in nature, in that the pressure between the cell and the buffer system will be maintained in equilibrium state actually, and it is non-toxic. Whenever you are using a buffer system, you have to keep in mind that it should properly maintain the buffer condition, that is pH range, pH buffering capacity that is within the culture. It should also be non-toxic in nature, that is it should not affect the cell viability, and it should coincide with the experiment that we will be doing. Suppose that we are using a buffer that might affect any other chemical reaction that we are providing within next experiment, then it will hamper our experiment, right? So, we need to first find out a buffer that does not cause all these problems. So PBS is isotonic as well as non-toxic to cell. That's why one is important thing, that is why it is used as a buffer in case of tissue culture, and also in case of washing as well as trypsinization. Trypsinization is something where you need to separate out the individual cell from the cell culture, so you can put it in a culture plate which contains an extracellular matrix and leave it in an incubator for it to grow. So trypsinization is a process that you will come to know afterwards also, and it should also support, PBS supports the cell viability by keeping the pH as well as osmolality in near physiological range, because the cell culture that you are taking from is from an animal or any other organism, right? So it should be maintained in the physiological condition there. So that should be kept in mind when you are providing or growing that particular cell in a particular cell media. So it should be in a near physiological condition. So you cannot completely mimic the physiological condition, because the physiological condition is always dynamic, it's never static, it always changes from one position to another based on the condition that you are providing. So what best we can do is that within that near range, within the near physiological range, you can provide and use a buffer which coincide with that particular physiological change also. So it consists of other components, as you have said. Sodium chloride is present other than the potassium chloride, sodium phosphate dibasic, and also potassium phosphate dibasic is there, which provides the phosphate ions that are necessary in this phosphate buffered saline. And sodium and potassium is also required to maintain the osmolality of cell. You can see that it is required to maintain the osmolality, because osmolality is something that keeps the viability of the cell intact, and you want it until your experiment is completed or the cell has been stored for a long time. You have to keep that in mind in each case. So that's why phosphate buffered saline or PBS is essential in cell culture, because it is an important buffer in this case. So anyone has any doubt in this case? Should I move into next question? Anyone has any doubt? Okay, so I will proceed into a next question then. So L15 media is used for dash free culture. So anyone knows the answer? Carbon dioxide is there, oxygen is there, nitrogen is there, hydrogen is there. So please provide the answer if anyone knows. Okay, I am getting the option A. So anyone else has any other answer? A. So majority of you, I think, put up the option A. So it's actually a carbon dioxide free culture. Basically, you can see that in the previous slide I have shown, right? Because I will just move back into a previous slide. Okay, so this particular system, right, this carbon dioxide, why is used as buffer there? It can also cause, it is important basically. It's an important buffering system or a gaseous buffer that is present. But the level in which this particular buffer system actually fluctuates is much higher than as compared to other buffer systems that are present recently, because carbon dioxide level of carbon dioxide dictates the pH range of this particular cell culturing system or the buffering system. So higher the the level of carbon dioxide might change, and it fluctuates drastically. So one of the disadvantage of this buffering system is the buff fluctuations are much higher as compared to other buffering system. So I will move on to this slide. So L15 media was discovered by this particular scientist, the Leibowitz, is a buffered media used for cell culture, cell growth and viability, and it does not consist of this carbon dioxide. Apart from that, it consists of other salts like sodium chloride, calcium chloride, etc. Other salts are also present there. Other free base amino acid, especially the essential amino acid. Essential amino acids are something that you need to provide from outside, right? Cell or the cell might be able to produce some of the amino acids which are called within the body itself, which are called as non-essential amino acid, while other amino acids are there, essential amino acid, which needs to be provided from outside. So apart from the amino acids, galactose is also there, which helps to maintain physiological pH control. So because of the absence of this carbon dioxide, you can limit the pH fluctuations there. Because the bicarbonic acid formation is less, and the proton formation is also will be less in this case. So you can limit the buffer fluctuation there. So that's why this media is consisting of carbon dioxide free culture. And apart from that, it consists of about L-glutamine, and it does not have sodium bicarbonate, because sodium bicarbonate is source of this carbonic acid. So it doesn't have that HCO3 minus that is provided within the media itself. And also in the absence of an incubator, CO2 incubator, because you don't need to provide any CO2 there because it lacks the sodium bicarbonate there. So and it can so the maintenance is quite less cumbersome as to what when we are using when carbon dioxide is used as a pH buffering control there. So I think hopefully this question is sort out. So anyone has any doubt regarding this? Any other question or any other doubt regarding this particular media, or why it doesn't have any carbon dioxide, or should I re-explain this for your additional knowledge and all? Okay, so I think this particular question is clear, so I will move on to the next question. So which of these features decrease after every passage in a cell line? So option is size, nucleus size, mitochondrial size, cytoplasmic volume, as well as telomere length. Which would be the answer? Okay, option D I am getting. Any other answer to this question? Okay, so I think everyone is sort of with option D, length of the telomere. That is the right answer basically. So basically telomere is something that is present at the end of the chromatis, right? And the genomic DNA is usually linear, right? So the ends are probably free. So this particular telomere is found at the particular end of this particular region. From this diagram also you can see that this particular sequence is a telomeric region. So from the slides that you might have seen in the cell culture technology class and all, you can see that with each subsequent cell division cycle, the length of telomere gets decreased because the DNA is linear in nature. So chances are that the exonuclease activity will be there, or a particular enzyme that regulates the length of this telomere, which is often called as you can see from this diagram, right? Telomerase, which actually extends the length of telomere so that the DNA won't get degraded. The efficiency of the telomerase might be coming down to a low point, or might be the chance that within each cell division cycle, the telomere length of the telomere will get drastically reduced within each cell division cycle. So when you are putting up a stock or a cell culture stock within -80, and then you are reviving it out, and then you are growing it again and again and again. So the cell is undergoing rapid division of cell cycle, right? It is undergoing multiple division cycle. So within each division cycle, chances are that this telomere end will get degraded over time. This will get degraded. So some of the regulatory control that is associated with this particular genomic DNA will also be affected, or more probably will be decreased, and this will lead to several other issues, including there will be changes in cell viability, cell growth, other phenotypic changes that might be changing within this particular because of this particular disruption or shortening of this telomere end, right? So whenever you are using that, you need to keep that in mind, because from year to year we are using different kind of research purposes, right? So if that particular phenotype is getting changed, because phenotype is derived, the cell phenotype is derived from the genetic material itself, right, which is obviously the DNA or the chromosomes. So if any change is happening within that particular genomic constitution itself, it will obviously lead to change in phenotype. So so the answer to this question is length of telomere. So the length of telomere decreases after every passage in a cell line. So so you need to do several techniques like karyotyping or DNA barcoding, etc., to see if any change is there within the initial cell line that we have acquired in a lab to the final stage that we are using, final stage of the cell line that we are probably using. So I hope the answer to this question is clear. So anyone has any doubt, or should I move on to the next slide?
No, sir.
Okay, so I think it's clear. So these are some of the basic questions that have been asked in the in the assignment section. So coming on to the next phase, next question, sorry. In which phase of the cell cycle two individual chromatids which make up the chromosome get segregated into each cell type? First M phase, G1 phase, S phase, or G2 phase. So anyone knows the answer to a question? Okay, M phase is there. I got an option A. Anyone has any other question answer? option D I am also getting. Okay. So I will clarify that out basically. I think in case. So the answer to this question is the M phase. So I am just popping out the same diagram that I have shown you earlier. So basically cell division consists, I will just clarify this one more again because I got an answer G2 phase here. So it consists of 4 phases: mitotic phase, G1 phase, S phase, G2 phase. So mitotic phase consists of these 5 different subsections, that is prophase, metaphase, anaphase, telophase and cytokinesis. This is the stage where the cell divides. So in the prophase the chromosomes are condensed together. In the metaphase as well as anaphase and telophase the chromatids are formed. The chromosomes are formed there. The chromatosomes are attached to each other by at the end of the chromatids, and later in the cytokinesis phase the chromatids get separated and cell also divides within each case, right, in the M phase itself. So that actually leads to the cell division there, because the chromosomes are getting later generated within the S phase which pass through a G2 phase, then it moves on to the mitotic phase there. The chromatids are also already present there, then which get condensed and then chromatids are formed, and later these chromosomes get separated and then cytokinesis occur. So one chromosome goes into a one progeny cell and the other chromosome goes into other progeny cell. So cell division takes place in this case. Afterwards it goes into G1 phase and then into G0 phase or S phase as necessary, as I have earlier said, and also in case of this particular and afterwards it might go into the, after completing into S phase it might go into a G2 phase. So G2 phase is actually called as a preparatory phase. It's not actually the stage where cell division takes place. So what happens in G2 phase is that the cell actually produces proteins. So it when it enters into mitosis it should undergo prophase, metaphase, anaphase, telophase and cytokinesis, right. So there are several proteins, several different proteins that are acting within each stage of mitosis. So in order to, before the cell has to enter into the M phase, it should have that particular raw material, that are these proteins or regulatory control substances that are needed within the cell. So the cell prepares all these regulatory contents within the G2 phase, and once this particular phase is completed, where the components are actually formed within this case, it then there are several checkpoints that are present at this particular stage, intermediary stage. So okay, several checkpoints are present within the intermediate stage. So once these components are passed, the cell checks itself whether these components are there, and once it has cross-checked itself, then or it has made itself correct, then it moves into the M phase. So it's actually the preparatory phase for mitosis, G2 phase, and M phase is actually the phase where the cell division takes place. So I think that particular explanation is correct and is quite understandable. So anyone has any doubt regarding this? So anyone has any doubt?
No, sir.
Okay, okay, thank you. So I will just move into the next question. So this is in basic question. So hepatocytes are cells that are found from stomach, brain, liver, kidney. Answer to this question, please.
Liver.
Liver. So this is a basic question, right? So I just put up a diagram for the liver cells there. These are also called as hepatocyte, liver cells. Hepato means liver and cyte means cell, right? So okay. So in case of brain, nervous tissue is there, and in case of kidney also there are different types of cells, and stomach is actually a combination of different type of cells basically. So I will just move on to the next question. So which of these properties can be determined using a cell counter? First one is exact density of an individual cell, cell surface property, quantitative growth kinetics, or size of DNA. So please, could you please give an answer to this question? First question is A, one is A. Okay. C is there. Any other answer? Okay. I am going to go with quantitative growth kinetics, because first I will go with the option A. because exact density, if I won't be more comfortable with this answer, because if it is like exact concentration of exact density of an individual cell, we cannot actually go with. But if that option will be like exact concentration of the cell density or the cell population, then cell counter would have been this particular answer, option A could have been also an answer. So self-prefer property is also not an application of a cell counter. And quantitative growth kinetics. So can you, one of you, please explain what is a growth kinetics is? So what all phases when a cell is growing, or when a cell is getting incubated in a particular temperature, when cell is growing, what all phases do it go through? There are different kinds of phases, right? So okay, so I will just show you the phase. So this is the growth kinetic curve, right? So first of all, when a cell is growing, it is reluctant to grow because it has to acclimatize to that particular condition that you are providing. So it goes through a lag phase, right? Then it starts to go up, the cell density, lag phase, log phase, stationary, and death phase, that's right. And so it goes into a growth phase, right? So this particular phase is called as a log phase because the cell grows exponentially. It's not logarithmic, it's kind of exponential. So one cell divides into 2. So if there are 4 cells, it divides into 8 cells, then each individual cell will grow exponentially. So it's like a pyramid actually, if you consider from a single cell. So that actually goes through a growth phase. Then it slows down at a particular range, because if we are keeping it a confined media, the cells might have used up all the components that are present within the media itself. So it cannot utilize any other components from externally because the media is already depleted. So the cell begins to die out. Before dying out, the growth rate actually bends down so that it becomes a stationary value. So that particular phase is called as a stationary phase. Afterwards, the cell is not growing. All the cells are getting old. So once it has reached that phase, the cell starts to die out. So basically, this is a growth kinetic curve. So in microbiology and all, you might have seen this particular curve, right? So you can actually see this growth kinetics. How? Because cell counter is an instrument that actually measures the concentration, or actually determines the concentration of a cell that is found within a particular culture. It's like a spectroplurometer. Hemocytometer is also a cell counter, because you might have seen a hemocytometer when you are doing practical, right? You put some amount of cell suspension into that, you put a slide which consists of a grid line like that, right? There's a grid line like that. From in each square you are counting the number of cells, and from the average number you are taking, you can determine how many cell actually is there. So it's also a kind of a cell counter where you have to do manually. So there are also other cell counters which is automated in nature. So based on the concentration of cell, you can actually calculate the growth kinetics. You can see whether the cells are in lag phase. If the cell OD is not changing, then it actually uses optical density or any other electrical impedance or any other mechanism to calculate the concentration of cell. So if the OD is not changing, it might be in lag phase. If OD is changing, then it will be in growth phase. Then after sometimes you can say that the growth is not no more happening, cells are not getting divided, so it's in stationary phase, and then the OD is coming down, then the concentration is coming down, meaning that the cells are entering into a death phase. So that's why cell counter actually measures the concentration of the cell. So you can basically determine the quantitative growth kinetics in this case. So I hope that is clear. So anyone has any doubt regarding this? So if anyone has any doubt, you can just ask, or should I move on to the next question? Okay, so I think I will move on to the next question then. So this is also a question: which of these does not lie under the biological safety hazards? First one: genetic manipulation, radioisotope in sterile cabinet, propagation of viruses, position and maintenance of laminar flow hood. So does anyone have any specific answer to this question? So you can just type the answer out, or you can unmute yourself. Does not lie, propagation of viruses, like so. So I will just go into one of the option, propagation of viruses. So basically, some might be like disease causing in humans, right? Infectious agents in right. So basically, this poses the question actually says that does not lie under the biological safe hazard. This actually lie under the biological safe hazard because the viruses that you are growing, we might not know whether it is harmful to us or not. So if it is getting propagated or it might have gone out of control, then that is actually a biological safety hazard. So the option might not be C. So position and maintenance of laminar hood, flow hood, that actually goes up to a particular answer because laminar hood can be used in different BSLs, right? BSL1, BSL2, BSL3. So basically, the maintenance of laminar hood and position might be a probable answer for that. And radioisotope in sterile cabinet, that is also a probable answer because radioisotope needs to be stored in perfect safe conditions so that it does not cause any radiation release or anything. So basic preferable bottles or any other cabinet should be there, not just a sterile cabinet basically. So this might also be an answer to that question. Option B can also be an answer along with option D. So that just an assumption. So anyone has any doubt to this question? Any doubt to this particular question, or is it clear? I will move on to the next question then. Okay, I will move on to the next question. I think that's clear. Some doubt is there basically on that answer. So in culture system, osmolarity of medium is measured by which option? What do you think? Measuring volume of solution, measuring pH, by titration, by depression of freezing point. Okay, I got an option D. Any other answer? Okay, again an option D. So I think this is an like something that you might have learned in +2, right? +2 or +1, I am not sure. So basically, the answer is by depression of freezing point, because this osmolarity actually is measured based on some properties called as colligative properties. Colligative properties are properties where a concentration of a solution can be measured based on the number of solutes that are present in that particular solution. So different properties are there that gives a direct understanding of the concentration of solute that are present within a particular solution itself. So the answer to this question is by depression of freezing point. So basically, if I give an extra point to this, basically osmolarity of a culture media is primarily measured by depression of freezing point. Okay, so because the number of solute actually that is present within the within that particular solution might affect the freezing point of that solution, because as the concentration of that solute or the number of solute particle within that solution increases, the freezing point of that solution also decreases. So there are different interactions that are happening within a homogeneous solution, right? When you are adding a solute into that homogeneous solution, it is kind of a heterogeneous solution because you are adding something from outside which is not present in that particular homogeneous solution itself. So it's a heterogeneous solution. So it's no longer pure. You are added something that is from outside to that solution, just to look at what it happens in this case. So basically, if you are adding more and more concentration of solutes or more and more number of solute particles from the outside into a solution, the freezing points gets decreased. It moves into a more negative value. Decrease means that it moves into a more negative value, right? So based on the freezing point, you can actually get some idea about the osmolarity of that particular solution. And there are also some instruments called as osmometers, which can actually determine the freezing point of solution. And if we know the osmolarity, then chances are that we can also determine the osmolality. So I think this answer to this question is quite clear. That is, in culture system, osmolarity of a medium is measured by depression of freezing point. So does anyone has any doubt? It's a type of a colligative property. So anyone has any doubt regarding this? So I think that is quite clear. So I will just move on to the next question. So bubble formation, the bubble formation is prevented in culture system because of shear stress to cell, changes color of media, changes temperature of media, and causes contamination. Okay, I got an option A to this option A. Another one I also got. Any other option? Anyone might have marked any other option? Option
Sir, A.
A. Okay, so the answer to this question is shear stress to a cell. So basically, when you are putting up a cell in a culture, right? So it should be maintained in a static condition there. The condition should be maintained dynamically, because a perfect cell culture is not there. It should be in dynamic, but some of the conditions should be not be present there. Especially one of the condition is shear, this bubble formation, because bubble formation actually exert a pressure within the cell culture system, and this might cause the cell to actually shear off or cause any change within the displacement within the cell or any other thing that might affect the cell viability and all. So bubble formation should be taken up in utmost priority and all. So and apart from that, other options don't seem to like coincide with the other, including changes of color media, changing temperature of media causing contamination. So the answer to this question is shear stress to the cell. So do anyone has any doubt regarding to this cell? Because bubble formation actually causes a pressure within the culture system, so we probably don't want that. So anyone has any doubt? So in the overall section, do you have any doubt? Like in the whole presentation that you are proceeding, does anyone have any doubt regarding how to if any additional points should be added or anything? So okay, so I think everyone is quite clear about the assignment section, the solution section that is being taken today. So
Yes, sir.
hopefully I think I will just note down the attendance basically, so please don't go anywhere. So okay, 37 attendance is there. So I think hopefully everything is clear or understandable. So I think we will all meet in the next week, Sunday at this time point itself. So thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.